Hey everyone, Dr. Hinkey here, and this is our video lecture for Chapter 6 on Metabolism, Energy, and Enzymes. This chapter is going to feel a little choppy when we first go through it. It's going to feel like we're jumping around between some unrelated concepts, but they will all come together because they're all parts of cell respiration and photosynthesis. So we need to understand all of these pieces together, and then they're going to roll up and we'll see, okay, I get this when we talk about metabolism, about how cells get and use energy. So the chapter will cover cells and the flow of energy. So we'll just have a general discussion kind of about energy. So we're looking at the physics. We've looked at the chemistry side of cells. Now we're going to look at the physics a little bit with the laws of thermodynamics. And then we'll look at metabolic reactions. So back to some chemistry and the interaction of chemistry and physics. And then enzymes, which you have already looked at in lab. So this chapter will be very familiar, that portion of the chapter. And then some chemical reactions that are going to be really important because we're going to have sort of a chain of these when we get to cell respiration and photosynthesis in our electron transport chain. So first, when we talk about cells and the flow of energy, cells' energy is the ability to do work. So in order for cells to do work, to do anything, we need to have some energy. And we have different types of energy depending on what we are going to use it for. Broadly, we have kinetic energy, which is the energy of motion, mechanical energy or movement. Walking is mechanical energy, uses mechanical energy. Energy of motion is water going over an waterfall, and we can capture that and convert it to electrical energy. Solar energy comes in through the sun, through light. And then we have potential energy, which is stored. It has the potential to do work when we release it. So water behind a dam is an example of potential energy or stored energy. Chemical energy, the energy contained in those bonds between molecules, or between atoms in a molecule, those bonds, those electrons moving back and forth, contain energy, and when we break those bonds, we release that energy. So while it's tied up in that molecule, in the food we eat, and carbohydrates or lipids or those are big ones for energy, but protein, any of those, we can break down those bonds to release energy. And that is potential energy. Once we've broken it down, it becomes kinetic, we capture it and we do something with it. When we look at the flow of energy, energy flows through systems. So we talked about energy flowing through systems versus matter recycling in systems. We looked at that a little bit in our first unit covering ecology, about how matter, carbon, is captured by plants as carbon dioxide. It's converted to plant matter, to biomass, which the biomass is carbs, lipids, proteins, nucleic acids, which are then eaten by other animals and converted to other carbs, proteins, lipids, nucleic acids, to our schnapps. And that schnapps continues to get converted every time we move up that trophic level in the food chain. When an organism dies, it decomposes, it releases all that schnapps back to the environment to be taken up by plants again. So nutrients cycle through systems, but our energy flows through. It means it flows in all energy in our, on the planet. So in our enclosed system, all energy comes from solar energy, from the sun. So solar energy starts it, brings the initial energy in that is captured by chlorophyll in plants, and used to create biomass. And when it converts, we lose some of that energy as heat. When the plant converts that to biomass, some is lost as heat. So the temperature of an organism is a measure of that heat that's being lost, that energy that's being lost as heat. So heat is a form of energy, We'll talk about that a little bit more in a second. When this moose comes along and eats the plant, it takes some of that energy. It uses some to walk. It uses some to increase biomass. But the temperature of that moose, it is radiating heat. It is losing some of that energy it took in as heat. So energy flows because eventually all the energy is going to be lost by heat. We're going to have a resupply. We're going to have to constantly have some flowing in because eventually it will all dissipate in the unusable form of energy of heat. So that brings us to our 2 laws of thermodynamics. The first law is called the law of conservation of energy. Energy cannot be created or destroyed, but it can change from one form to another. So we just saw that in that image of solar energy coming in, being captured by plants and being converted to chemical energy in the bonds of carbohydrates, lipids, proteins, and nucleic acid. Some of that energy is used to grow, so as mechanical energy, and then some is dissipated as heat. So heat is still a form of energy. We're not destroying that energy. We're just changing form. And heat is one of the forms I can change it into. The second law of thermodynamics is called the law of entropy. Entropy is another word for chaos. And this law tells us that everything goes to chaos. When energy is changed from one form to another, there is always some energy that is going to be converted or lost as heat. It will be converted to heat energy, and we cannot capture that. It dissipates too quickly to be captured and used to do work. Remember, the energy is the ability to do work. Heat dissipates too quickly. I could capture it. I could condense it back and do work with it, but it would take more energy to capture it back to a form that I could use than I would get out of it. And so we say it's lost. So, and just some examples of that, that we're all aware of, but maybe just put it together a little bit. Your car, after you have been running. So your car takes the chemical energy, the potential chemical energy stored in gasoline, in those carbon-hydrogen bonds of gasoline, and it converts it to mechanical energy, and your car moves. So everybody's gotten out of the car after a ride and put your hand on the hood and it's hot. It's because while we're converting that gasoline, the potential energy to kinetic energy, the energy of motion, we lose some as heat. So cars, really fuel efficient cars might be 60% efficient. So for every $1 you put into your gas tank of gasoline, 60% might go to moving your car forward and 40% goes to heat. Your water heater, hot water, if you have an electric hot water heater or a gas hot water heater, you're paying an electric bill to put electricity in to convert from electrical energy to heat the water. That electrical energy is used to heat the coils that heat the water. If you go and stand next to your hot water heater, put your hand on it, it's really, really hot. You step away 6 inches, it's warm, but not hot. You pull your hand about a foot away and it's cooler still. That shows how quickly that energy is lost. So even though we'll insulate that hot water heater to keep that heat there as long as possible, we have to keep adding heat. Otherwise that water will get cold because that heat is dissipating. So heat dissipates really quickly. No system is 100% efficient. If it was, for every $1 we paid on the electrical bill, would be converted to 100% heat for that hot water heater, and it would stay hot. We'd never have to heat it again. But that heat dissipates, so we lose heat. So the new light bulbs, we used to have the incandescent light bulbs that were really, really hot. If you remember those old light bulbs, if you still have those in your house, if you turn the light off, you really can't go and grab and unscrew that light bulb because it's so hot, because about 90 to 95% of the energy that went into it, the electrical energy, came off as heat. and only between 5 and 10% was actually converted to light energy. So for every dollar of your electric bill spent on turning on one of those light bulbs, 90% went to generating heat. So that's what we mean by we lose it. We lose energy to heat. It dissipates really, really quickly, and it's not used to do the work we want it to do. So here's our laws of thermodynamic. As a picture, sun, our solar energy comes in. It is captured by a plant, and it is converted to plant mass, to carbohydrates. So that is potential energy. It's stored energy. And some is lost as heat. Then an animal can come along and eat it and convert this stored energy to motion, to movement, to growth, to reproduction, to biomass, and some is lost as heat. When we eat food, we eat carbohydrates. This is our potential energy, lots of energy stored in those carbon-hydrogen bonds and carbohydrates. We use it to do some work, like move, which contracts a muscle. And as you all know, if you've ever worked out really hard, the harder you work out, the more work you do, the hotter you get. You generate heat, you release heat from doing work. So all of those cells, remember metabolism, the sum total of all the chemical reactions in a body. Those chemical reactions are, many of them, energy transformations. We are going to transform energy from one form to another. I'm going to transform it from one type of chemical energy to another, from chemical energy to biomass, from chemical energy to kinetic energy when I move. So we're transforming those. That means, based on the second law of thermodynamics, where I'm going to constantly be losing some energy to heat, that means I just always have to keep resupplying my energy. I have to eat. Right? Plants have to constantly have solar energy input so that they can make their chemical energy, their glucose. So metabolism, the sum of all those chemical reactions. And we talk about chemical reactions. We have the reactants, the things going in, and the products, what comes out as a result of that chemical reaction. So I draw that with an arrow going from the reactants, what goes in, pointing to the products, what comes out. And free energy is the amount of energy that's available to perform work. So free energy, how much energy is there available right now to do work, where I don't have to do any conversions to release energy. And I can have chemical reactions. Some chemical reactions are exergonic. The end products have less energy than the reactants. They release energy. These occur spontaneously. These are typically taking large products and having them break down to smaller products. They release energy as they break down. As I take a big molecule and I break it to smaller molecules, I release energy. If I'm going to build something, if I'm going to take two small molecules and put them together, think about building blocks. It takes a lot less energy to kick a bunch of blocks over than it does to actually stack those and put them together. So endergonic, endo, into, exo, out of. Endergonic reactions require energy input. Erg is a measure of energy, so ergonic, endergonic, energy into. These are non-spontaneous. I need to get energy and put energy into the process to do this. Where do my cells get energy from? From ATP. And when we talked about our macromolecules, we talked about adenosine triphosphate. Adenosine triphosphate is a nucleic acid. It's a single nucleotide made of ribose and adenine. That's my adeno on that side, adenosine. And then triphosphate. 3 phosphates attached to it. It's a high energy compound, and we'll talk about why when we look at a picture of this, but it's constantly being broken down. We use it to drive chemical reactions. And so what we'll look at with our ATP is that we always use that as coupled reactions. When our cells do work, we always have a coupled reaction. That means we have to have an exergonic reaction coupled with an endergonic reaction. In our cells, in order to do work, we need energy. The energy my cells use comes from breaking off that third phosphate from my ATP molecule. So I have lots and lots of energy stored here because this is kind of unstable. This third phosphate really doesn't want to be here. So it breaks off easily, and when it breaks, that chemical bond releases energy that my cell can use, can capture and use to do work. We'll get into this more when we talk about cellular respiration, but a lot of times people say, well, wait, when we learned the macromolecules, we learned that glucose is the cell's energy source. That's how my cells use energy, is from glucose and carbohydrates, and stores fats and uses those. So cellular respiration, what we're going to look at is how we go from my glucose, why do I have ATP? And the analogy you're going to see in one of the videos I've posted, and that I'll tell you about here, because it helps. Because we're saying, well, what's with this intermediate molecule? What's this ATP? I have glucose, I'm good. If we think of going to the laundromat, if we think of energy as money, if I go to the laundromat with a $10 bill, can you get your laundry done? No, right? Because the machines don't take a $10 bill. First, I have to go to the change machine and break my $10 bill into quarters. Now I have 40 quarters. I can go over to the washing machine and plug my quarters in, and the washing machine will work. Won't work if I try stuffing that $10 bill in. So glucose is my $10 bill. Glucose is great. It's got lots and lots of energy, but my cells can't accept that. So the first thing I have to do is take my glucose to the mitochondria, that's my change machine, put my glucose in, and get my ATPs, my 36 to 38 ATPs out. And then the ATP is like the quarter. That can work in the cell's machinery, so the cell can do work with ATP. So that's why I have this intermediate molecule. Glucose is the $10 bill. So the machine doesn't turn my $10 bill into 40 quarters. The $10 bill still exists. It's in the machine, and the machine gives me quarters back. Same thing, I am not converting my glucose into ATP. I'm not rearranging the carbon, hydrogen, oxygen, and somehow picking up phosphates to make this molecule. I'm putting in the glucose. My mitochondria, my change machine, breaks down all the chemical bonds in the glucose, captures that energy to build ATPs. What's it building ATP from? Well, from adenosine diphosphate. So our bodies are really, really good recyclers. We're very efficient. Remember it said, our energy efficiency, 100% of the energy that goes in most mechanical systems, most man-made systems, if we get 20, 30% efficiency, we're just doing great. The human body recycles a lot of things. We're very efficient, closer to 60 to 70, which doesn't sound like a lot, but compared to man-made systems, we're really great. But one of the things we recycle and reuse are our ATP molecules. And what we'll do is break off this P. Remember I said this is unstable? I break this third phosphate off, and that breaks that ATP and releases energy. That's an exergonic reaction. Breaking off that phosphate, going from a big molecule to two smaller molecules, adenosine diphosphate and phosphate, releases energy. That's what my cell uses to do work. Now I have these two molecules. What I do is in cell respiration, I break down my glucose, and when I break carbon-hydrogen bonds in my glucose, it releases energy that I use to put that phosphate back on to my ADP, resulting in ATP again. So I'm ready to go do that again and break off that phosphate, get energy to do work, and then use some of the energy from breaking down my glucose to put my phosphate back onto my ADP. So it's this continuous cycle. And just like with our laws of thermodynamics, I have to constantly be putting in more energy. I have to constantly be putting in more glucose. I always have to eat. So we call these coupled reactions because when I break down a big molecule and release energy, I'm doing that to use that energy for something else. I capture that energy, I use it. So the coupling we're looking at is in my cells, I use the energy that's released when I break the carbon-hydrogen bonds in glucose during cell respiration. That released energy is used to join a phosphate onto ADP to give me ATP. That's my energy currency for my cell. That's the molecule my cell can use to do work. And the way it does that is by breaking off that third phosphate and releasing energy and doing work. So if we look at a specific action, so myosin for my muscles to contract, when my muscles relaxed, there's an ATP sitting here on the end of my myosin fibers. Everything's relaxed, nothing's moving. When I move, I'm going to break off. That phosphate breaks off, releases energy, and that energy is turned to mechanical energy of movement. My myosin changes shape, grabs the actin, and pulls it. Right? So my muscle contracts by all those actin and myosin fibers moving past each other, and that allows movement. But now I have an ADP and a P. And when that falls off, well, it makes room, and my next ATP comes in and bonds, and my muscle relaxes and waits till I need to contract again. And when I need to contract again, I need that energy, so I break that phosphate off. But that ATP comes in again because I just ate something and I used that glucose to make new ATP. So we have these coupled reactions. All right, so those are my coupled reactions. That's ATP, and we're going to look at that specific, the process we use to generate ATP from ADP plus P in cellular respiration. The other concept we need to put together to understand what's going on in cell respiration and in photosynthesis are how enzymes are involved in metabolic pathways. So typically in our cell, when we talk about metabolism, all the chemical reactions that take place, we see, I start with this, I get this. I start with glucose, C6H12O6, and oxygen, 6O2s. And then at the end of cell respiration, I get 6 carbon dioxides and 6 waters. We all learned that equation in school for cellular respiration, and it looks like it's a single step, but really it's a metabolic pathway. There are about 72 individual steps in between. So that one arrow that I see going from glucose and oxygen to carbon dioxide and water is really 72 little arrows, more similar to this pathway. I start with my reactants, and I make a change, and I make another change, and another reaction, another reaction, another reaction, till I finally get to my end products. So we have all these intermediates in here that we don't always show in my chemical equation. So this is called a metabolic pathway, and I have all these intermediate steps. And remember, the sum total of chemical reactions in a cell is metabolism. Every single chemical reaction in our cells is catalyzed, it's sped up by an enzyme. And we need enzymes to speed up those chemical reactions because when we need the products of a reaction, we need them now. We can't wait for whenever its old sweet time gets around to doing that reaction, which would depend on those two molecules getting close enough to react. We need it now. So we need to speed up chemical reactions, and enzymes will speed reactions up to 1 billion times faster than they would occur if we let them go at their own rate. Enzymes can't make a reaction happen that wouldn't happen anyway. They only speed up a reaction. So our enzymes are protein molecules. That word catalyst means they speed up reactions. When I'm looking at a chemical reaction, those molecules that are on the left side of the equation with an arrow facing right are reactants. What comes out are products. When an enzyme's involved, we can also use the word substrate for the reactants. That's a synonym when an enzyme's involved. So each chemical reaction has a specific enzyme. So remember, our proteins have a very distinct shape. That primary structure of amino acids bends and folds and twists in a very specific way through the secondary, tertiary, and quaternary shape. That shape is specific to the job it does. So to the molecule it's going to act on. That's why it's so important that we have the right shape. So each metabolic pathway, each step in my path requires a different enzyme, because going from my reactant A to B, I'm going to change A into B. So here enzyme 1 does that. Well, enzyme 1 can no longer work on B because it's the specific shape for molecule A. Now I need an enzyme that's the right shape to grab onto molecule B to convert it to C. So enzyme 2 comes in, which is the exact right shape for B and can perform the reaction B to C. Now C is my substrate or my reactant. I want to convert it to D. Well, enzyme 2 here is specific to B, so it can't change C. So enzyme 3 comes in. So each step has a specific enzyme associated with it. The way my enzymes work is they form these complexes. They bind, their specific shape is appropriate to their substrate. They bind to that, and when they bind, it causes a slight conformational change in shape of my enzyme that makes that chemical reaction happen, and then it releases the product. We call this the induced fit model. So if we induce something, we make it happen. The chemical reaction is induced, it happens when that substrate fits into the active site of my enzyme. So I have different enzymes for different reactions, and I have enzymes that can both break down or build molecules. So I can do those exergonic reactions or endergonic reactions. Either one, even though the exergonic reactions would happen spontaneously, they might not happen fast enough. When my cell needs it, it needs it. So I need my enzyme to speed that up. So here's my enzyme. This is my substrate. If I'm looking at a degradation reaction, I am going to break down a big molecule into smaller molecules. My enzyme has an active site that's the exact shape for the substrate to fit into. Once it fits into that position, this is the active site. When the substrate binds to the active site, I now have the enzyme substrate complex. The enzyme is going to change shape just slightly to make the chemical reaction happen quicker, and it's going to release the products. I went from one big molecule to 2 small molecules. Those are my products. That's done. The enzyme takes off. So the enzyme's not involved in this reaction other than makes it happen, off it goes. So when I write the chemical reaction, it would be glucose plus oxygen. The glucose comes into the enzyme, breaks down. It would be 72 steps. And what I'm going to release, carbon dioxide and water, smaller molecules. That enzyme is not written in that equation anywhere because it's just a helper. So my synthesis reaction, you could get my analogy for this. My synthesis reaction, my enzyme is a specific shape that would let these two molecules come in. I'm going to build a big molecule from smaller molecules. They attach at the active site. I get a slight conformational change. That induced fit makes the reaction happen that joins these two molecules into a larger product. And off the enzyme goes. For these synthesis reactions, I think of the enzyme and say it's not involved in the reaction as being a yenta, a matchmaker. It's the matchmaker. It goes and it finds these two substrates and it hooks them up and off they go happily ever after, get married, get joined together, and the yenta goes off to do that to some other substrate. My matchmaker heads off to do more matchmaking, doesn't stick around for the marriage, is not involved in the marriage, is not part of the marriage. So that's my enzyme analogy for a matchmaker for synthesis reactions. So the way my enzymes actually work, what they're really doing in order to speed that reaction is they're lowering the energy of activation. So my molecules in my cell wouldn't necessarily react with each other unless they're activated. A lot of times in my cells, what that has to do is with where they're located. They have to come into close enough contact for those valence electrons to interact for a chemical reaction to happen. So most chemical reactions need a little push. Even if it's an exergonic reaction, I have to give it a little push to get it started, to get it kick-started. I'll get more energy out than that little push to get in. So we say it's exergonic, it releases energy, but still needs a little push. We call that the energy of activation. Enzymes lower the energy of activation. They bring the substrates into contact with one another, or the substrate in the enzyme so it can break down that big substrate. So that's going to speed up the reaction, just pushes things together quicker. And if we wanted to map out that energy, my normal chemical reaction, even if I'm going to release energy, so I have a big high energy molecule, this is my free energy, and at the end when I break it apart, I release energy. Now I have two low energy products because I've released that energy. I'd still have to put some energy in to make this happen. And normally I would have to put this much energy going from here's the energy I have. I got to give it a little kickstart. And now I can, and now I can release all of this. So my net gain in energy is from here to here. That's how much energy was released in this reaction, but I had to put some in first. If I add an enzyme to this, I have to put in a lot less energy to kickstart it. And so that makes the reaction happen faster. All right, so how can we affect our rate of enzyme activity? This is what you looked at in the lab. Factors that affect the rate of enzyme activity, substrate concentration. Enzyme concentration can also change the rate of reaction, temperature, and pH. So let's look at each of these. The effective temperature on a reaction. We see at low temperatures, my reaction's slow. Well, cold temperatures, molecules move slower. So I'll have less reaction. Remember, the more my molecules bump into each other, the more I'm going to have reactions, the more my reactants or my substrate bump into the enzymes that are available, the faster the reaction will happen. And we see that as I increase temperature, look, my reaction happens faster and faster and faster and faster until this point here. This is my optimum temperature for this enzyme. And then it drops off really quickly. And the reason for that is at high temperatures, I start to break those hydrogen bonds that hold the protein in its very specific shape. So that secondary, tertiary, and quaternary level that's held together with hydrogen bonds that break at high temperatures, my enzyme loses its shape. When it loses its shape, it no longer has the right configuration on that active site to do its job, I denature my protein. As I get colder and colder, I don't denature my protein. I just slow the molecular movement. So I'm not going to denature on this end. So this can slow down until I get to absolute zero when I have no movement, which doesn't exist in the real world. But high temperatures denature it. And if you think about the egg white example for denaturing a protein, once I have my protein denatured in egg white, even if I cool it off, it's not going to go back to its original shape. My egg white's not going to turn transparent. So this destroys the protein, and that's the end of the reaction. The effects of pH, for most enzymes, you see human body temperature is 37 degrees Celsius. It's about right here. So if you think when I'm sick, my body resets my thermostat a little higher so I can move molecules faster. Part of that is to get my immune system cells to the infection. But it's dangerous. I can't go too high or have this temperature going on too high, or I'll break down my proteins, the enzymes that are doing some of this work here. pH, however, so there's not that optimal temperature for most enzymes, is just a little bit over 37 to 40 degrees, and then we start to break up. pH, it depends on the enzyme. Different enzymes have different happy places for pH. So depending on where the enzyme normally functions, so trypsin, which works in our small intestine, which is slightly basic, has its optimal function at a pH of around 8. Pepsin, which works in our stomach with our stomach acid pH of 2, has its optimal function, optimal rate of activity at a pH of 2. As I lower my pH, my ability of my enzyme to work drops off because those excess hydrogens interfere with the shape. Similarly, if I increase pH, those excess hydroxides interfere with the shape of my molecule, of my protein, the enzyme. But the optimal varies depending on where that enzyme is typically found. Where does it usually do its job? So we always think neutral is the best pH. Not necessarily. pH of 7 would be really bad in the stomach. So the enzymes are adapted to where they do their job and to the pH where they do their job. So with regard to pH, enzymes operate at an optimal pH based on where they do their job, what their job is. And if I get higher or lower, that rate drops off. The rate of enzyme activity slows until it stops completely when that enzyme denatures because of the interference of pH. So we think about homeostasis. Our bodies maintain homeostasis with regard to body temperature and with regard to the pH of different systems within our body. Most of that homeostasis comes back to keep our enzymes happy, because those enzymes control the chemical reactions in a body. That's metabolism. Without happy enzymes, our metabolism goes kaput. The third factor that we looked at in lab was concentration. So I can have some cell regulation of enzyme activity. If I don't need an enzyme, I don't make it. So cells can regulate the presence or the absence of an enzyme. They just won't make that enzyme. It's a protein. My DNA has instructions for how to build a protein. I don't need it. I'm not going to make it right now. Oh, now I need it. I'm going to make it. So we have regulatory mechanisms. Cells can regulate the concentration of the enzyme. If there is more of a substance that the enzyme acts on, the cell will make more of that enzyme so that it can work faster. Or, hey, you know what? There's not too much of this. An example from microbiology is E. coli. If lactose, which is a sugar found in milk, is present, they will make lactase, the enzyme that breaks it down. Normally, most environments, glucose is the predominant sugar, not lactose, so they won't make lactose. But if there is some lactose present, they can make it. They can make more. If there's more lactose, they need more. So they can regulate the concentration, or they can activate or deactivate them. They can do that through the production of cofactors. So some enzymes have a cofactor. It's something that must be present for the enzyme to do its job. And coenzymes are things that are non-proteins but interact with the enzyme to help it work more efficiently. So a lot of vitamins are coenzymes. And what a cofactor will do, this is my active site. If the cofactor is present, makes it easier for the substrate to bond. So when we talk about being able to activate enzymes or stop enzymes from acting, inhibit them, I can prevent an enzyme from doing its job when I don't need it. Maybe I've produced the enzyme, but now its substrate is gone, or I still need some of that substrate. I don't want the enzyme to continue with the reaction until the reaction goes to completion, meaning that all the substrate's gone. Maybe I want to have a low level of that substrate present, so I don't want the enzyme to work until all the substrate's gone, so I can inhibit it. And I can do that two ways. I can create substances that are enzyme inhibitors, and some of those may be competitive inhibitors and some are non-competitive. What they're competing for is the active site. So a competitive inhibition is when the molecule that inhibits or stops the enzyme from acting is the same shape as the substrate, so it binds to the active site. Once it's bound to the active site, enzyme can't do its job anymore. Non-competitive inhibition is when I have a product that binds to a different site. So a lot of times with non-competitive inhibition, the end product of the complete metabolic pathway is a specific shape for a different site on the enzyme, not the active site, but the allosteric site. Allosteric means far away. It's away from the active site, but this end product, hey, once I've made enough of this end product, I don't need this reaction to take place anymore. So once I have enough of this, it can come in and bind at that allosteric site away from the active site. And when it does that, it changes the shape of the active site from this, the shape of molecule A, my first reactant, to this. Oh, doesn't fit. I've now stopped the reaction without interfering or without taking up the active site, but with actually just changing its shape. All right, oxidation reduction. Here, we're jumping again. So this, I know it seems like we're jumping between unrelated concepts, but they are all part of my cell respiration. So the last chemical reaction we're going to talk about here is oxidation reduction reactions. Oxidation reduction reactions are often called redox reactions, and they're reactions that look at the movement of electrons between molecules, or between elements. So an oxidation reaction is when there is a loss of an electron. A molecule or an element loses an electron. It is oxidized. When a molecule gains an electron, it's reduced. And these take place at the same time. We've seen an example of this in our ionic bond. My sodium, with its lone 1 valence electron, gave that up. My sodium was oxidized. The chloride gained that electron. It was reduced. Right, so these two reactions take place. That was an example of forming an ionic bond, oxidation reduction reaction. These can take place among molecules, so not only elements but molecules. One of the molecules is going to accept electrons, the other is going to give it up. The book, I think, uses oil rig to remember it. Oxidation is a loss. Reduction is a gain. I always use Leo the lion goes grr. Leo, L-E-O, loss of electrons, oxidation. Grr, G-E-R, gain of electrons is reduction. So whichever one of those works for you, that's how to remember this. This reduction, gain of electrons, this trips people up a lot. Remember, if I gain an electron, I now have one more negative charge in an electron than I do positive charges in my nucleus with the protons. So my overall charge is negative. When I gain an electron, my overall charge is reduced. That's why we have these seemingly contradictory terms, is because an electron's charge is negative. So if I have more negatives, I'm taking away, I'm reducing. This all will come into play when we look at our next chapters for photosynthesis and cell respiration. Photosynthesis, I take two small molecules and I put them together to build a larger molecule, glucose, and release some waste product, oxygen. So this is endergonic. I have to put energy in order to build a bigger molecule from a small one. It uses lots and lots of enzymes because, as I said, this arrow here is really lots of separate small steps that take place. And in this process, this is also an oxidation reduction reaction. So oxidation, loss of electrons. My water is going to give up electrons to carbon dioxide. And when it does that, it becomes oxygen. My carbon dioxide is going to gain those electrons and become glucose. So I can say carbon dioxide is reduced to glucose. Water is oxidized to oxygen. And we'll look more closely at that in our photosynthesis discussion. Cell respiration, I take a big molecule, and there are two molecules involved in this, but glucose is my big one that I'm going to break down. So I'm going to break apart a big molecule into two smaller molecules, carbon dioxide and water. And I'm going to release energy in the process. It's exergonic. About 72 separate steps and enzymes involved. And this is an oxidation reduction reaction. Oxidation, loss of electrons. When I break down my glucose, it loses electrons. Glucose is oxidized to carbon dioxide. Those electrons are gained by oxygen. So oxygen is reduced to water. So we will zoom in on these in our next two chapters. We'll also take a closer look at the chloroplast and the structure. We talked about that briefly in chapter 4. We'll look in more detail. Same thing with the mitochondrion in chapter 8. And all those stepwise chemical reactions that we'll have taking place. These are really challenging chapters, so spend some time. You're going to need more than one look through the videos. You're going to need more than one read through the book. You're going to need all your resources, the videos and the book and the PowerPoints and the recorded lecture, and go through slowly. Break everything down stepwise. Learn one step really well before you move to the next. It's really the only way to tackle it. All right, so I should have a photosynthesis and cell respiration video. I should have those up by Thursday. So thank you all, and I will see you in the classroom.